Hello everyone. Let us take up a new topic today, that is drug discovery and clinical evaluation of the new drugs. Okay, so far we have covered the general pharmacokinetics and pharmacodynamic processes. Exactly what the pharmacology of a drug is, or how it can be determined, or what exactly happens to the drug when it is administered, or what the drug is doing to our body when it is administered. Also, we have covered the adverse drug reactions and the factors which are modifying the types of actions which are produced by the drugs. Now, let us see how exactly a new drug comes into market. So, to bring a particular drug into the market, what are those different processes which are involved in it? Okay, how exactly it happens? What are the basic steps which are involved in the drug discovery? Okay, and how exactly they are evaluated in human beings to determine the safety or the efficacy of that particular drug, whether it shows effectiveness or no. Okay, because you just can't take a particular chemical and call it as a drug. Okay, you need to carry out some systematic study about that particular drug as to how it interacts. We have seen all the general processes which are involved in the kinetics and dynamics. So all these needs to be studied about a drug. A detailed study has to be done about all these processes which are happening, exactly with what our drug is getting interacted with, what are those different processes which come into picture once the drug acts in our body, and how exactly the effects are executed, or whether it is effective against a particular condition or no. That has to be properly evaluated before it can be marketed, right? So several marketed preparations are available, right? So before any drug comes into the market, there are several steps which it has to undergo. Several studies are carried out, and then and only then the drug is released in the market. It's not easy to bring a particular drug into a market. Okay, so in this particular lecture we shall consider how exactly a new drug is developed. What are those different processes which are involved in a new drug development? Okay, so basically there are three main components of this process of new drug development. So you can call it as a project. Okay, individual drug. So likewise there may be several drugs which have to be studied for a particular condition. Supposing if you are developing a drug which is supposed to treat, say, diabetes, to bring down the blood glucose level. Okay, so there may be number of drugs which may be brought into market, but for that drug to be brought into market, okay, you need to identify exactly which drug can lower the blood glucose level effectively. Now, not only that, which are the drugs which are quite safe. We have seen several parameters which are studied in every pharmacokinetic and dynamic aspect. So, how well those parameters are satisfied by a particular drug. So, accordingly, a number of drugs may be discovered, but hardly few of them will be actually marketed, will be actually prescribed to the patients who are suffering from diabetes. Okay, so when you start with the drug discovery, you will have a number of drugs which could be able to lower the blood glucose level, but hardly any one of the drug might actually reach the market, and that too after years of the studies which are being carried out. Okay, so we will see why this particular process it leads to a rigorous knockout of several compounds which are initially showing effectiveness. Okay, so let us see how exactly a new drug is developed. So basically there are three components of this process of development of a new drug. So the first one is the drug discovery. So here you make a proper choice of certain candidate molecules. Okay, certain molecule, you can't call them a drug as such at this stage, because yet they have to be studied for that particular situation, right, or that particular condition. So you just can't call them a drug at this particular point. So it's just a chemical molecule which you are discovering, which are chosen based on their pharmacological properties. Okay, so what is that best chemical which could be effective against that particular situation or that particular pathological. The next step is the preclinical development. So preclinical development, in the sense, these involve the non-human studies which are done. So before anything could be introduced to the human beings, you just can't take any chemical which you are not sure of that it may treat a particular condition, or it is not tested for being safe. So that cannot be tried in the human beings unless and until you carry out some preliminary testing. Okay, so you need to be sure of whether it is safe or no for it to be used in the human beings. Okay, so there are a number of non-human studies which are being performed at this particular level. So testing like toxicity testing is done in the animals, or the pharmacokinetic pharmacodynamic analysis have to be done, or the formulation has to be developed. So a particular drug, it has to be administered to the human beings, right? So you need to develop a particular formulation into which it can be administered. Okay, so all these studies are involved under the preclinical development of that particular drug. Next step is the clinical development. So here the selected compounds are tested for their efficacy or their side effects or potentially dangerous effects, hazardous effects in the volunteers or the patients. Okay, maybe healthy volunteers or patients. So here you are actually administering the drugs to a selected population of the human beings. Okay, so when you assure the safety of the drug in the preclinical development, you go for the clinical development. So here also it is administered in the restricted number of individuals. So when you find it safe in preclinical development, that doesn't mean that the drug is now safe and it can be marketed. Before it can be marketed, it has to be studied in the human beings also in a limited number. So there are several phases into which it is studied in the human beings also, whether you have to select the healthy individuals or whether you have to test the drug in the pathological states in which it is supposed to be used. Okay, so these are the three main steps of a new drug development. So for it to come into market, for a drug to come into market, these are the several processes which have to be carried out step by step. Okay, so we shall see all the three steps in detail. So coming to the first one, that is drug discovery. Okay, so here you are discovering a new compound, okay, or maybe a new use of a particular compound which is already being used for some other condition. Okay, so basically here drug discovery indicates the several identification steps. Okay, so there are several steps which are supposed to be carried out as a drug is discovered. So you just can't take a chemical and you just can't declare that yes, it can be effective in lowering the blood glucose level or it can lower the blood pressure. So you need to identify several targets. You need to see how it interacts with that target. Okay, so nowadays there is a lot of advancement, biological advancement has been done. So each and every molecule, each and every biomolecules in our body has been studied in detail. It has been cloned. Okay, so you know the several targets in the body, okay, on which the drugs can act. We have also covered those different targets on which the drug can bind. Enzymes are there, transporters are there, receptors are there. Okay, so mostly they are protein in nature. Sometimes they are non-protein in nature also. So you need to identify first of all what is that target on which your said drug under study it can act. Okay, so these drug targets, these may be either proteins, mostly they are proteins, or they may be non-proteinous also sometimes. Okay, so in the olden days, the studies were directly carried out in vivo. Okay, that is, for example, drugs may be studied directly for prevention of the seizures, experimentally induced seizures, or maybe for lowering of the blood sugar, or some inflammatory response suppression was checked. Okay, so here it was not checked whether with what target your drug is binding to in the body. Okay, so in the olden days, but now the advancement is such great that first of all you need to identify what is your drug target, okay, onto which exactly your drug will act. Okay, so it's very important to study in detail which are the possible targets of your drug, okay, based on the mechanisms by which they could be altering the pathophysiology. Okay, so by knowing several biological processes or physiological processes, you can identify certain targets, or you can say that your drug might act on this particular enzyme, or your drug may act on a particular receptor. Okay, so you can have an idea from the basic physiology of the body. Now say for example, let us take one example. Now it is well known that inhibition of angiotensin converting enzyme, it lowers the blood pressure. Okay, ACE inhibitors, these are quite popularly used antihypertensive drug. Okay, so this angiotensin converting enzyme, its main function in the body is to convert angiotensin 1 to angiotensin 2. Okay, so it suppresses the formation of angiotensin 2, which is actually responsible for the pathophysiology of the hypertension. So this is one of your target, angiotensin converting enzyme. So this particular knowledge led to a development of a new target. Why not target the receptors on which these angiotensin 2 is acting? Okay, so angiotensin 2 receptors, if you block them, obviously it will serve the purpose, right? It will help in reducing the blood pressure, right? Okay, so since the synthesis inhibition is controlling the blood pressure, you can even block the receptor, so that will also serve the purpose, right? So if you develop the antagonist which can block these receptors, that will also produce the same kind of effect, right? So they will also tend to reduce the BP. So this is how it led to the development of the sartans. Okay, there are several drugs which are acting as ARVs, angiotensin receptor blockers. Losartan, these are quite popularly used drugs, losartan, candesartan, irbesartan, and so on. Okay, so a lot of drugs have been developed after that. Okay, several derivatives have come into market which can lower the blood pressure. Okay, similarly, knowledge about the pathophysiology of the breast cancer, so it involves the estrogen sensitivity. So this led to the discovery that aromatase inhibitors such as anastrozole, this could prevent the estrogen synthesis. So since estrogen is involved in the pathology of the breast cancer, why not inhibit the synthesis of this estrogen? So that could also be one of the target for the drug discovery. So you could develop several drugs which could inhibit this enzyme aromatase, because of which the estrogen synthesis could be affected. So that's how it will help in the development of new drugs for breast cancer. Okay, so this is how the first step in a drug discovery is to identify what are the targets onto which you want your drug to act, okay, or to interact with. So that will make easier, okay, for you to develop several new drugs. Okay, so you can select only those drugs which can selectively act on these targets. So that will rule out unnecessary synthesis of several drugs. Okay, so you, when you are target specific, that becomes it reduces the work, okay, on the part of the synthesis. Okay, rather than going on synthesizing the compounds in general, you can be target specific. So that will reduce the work. Okay, next step is the lead finding. Okay, so now you have a target onto which you know that if your drug acts on that target, if certain enzymes are inhibited or certain receptors are inhibited, you know that this is the outcome which is shown. Reduction in the BP maybe, or treatment of the cancer can be done. Okay, so you have your target now onto which your drug can act. Now the next step is to find your lead compound which is best suitable, which will act on that particular target. Okay, so here you focus on the few lead compounds which may be derived from the synthetic chemistry. Maybe you are synthesizing some compounds in the laboratory. Okay, so you can target your synthesis, or you can plan your synthesis in such a way to develop those compounds which can interact with the target which you have identified. Okay, so the target molecule in your body which you have identified, so you can synthesize certain compounds with the help of synthetic chemistry which can selectively bind to and act on those targets. Okay, so here the several targets have been identified, they have been cloned, their sequence has been studied. Okay, mostly they are proteins, so their sequence have been studied, the amino acid sequence have been studied. Okay, so by this procedure you know what is the structure of your target. Okay, specifically you should go for the human forms because there may be variations in your drug action depending upon the species. We have seen that species is one of the factors which can modify the drug action, right? Okay, so you should be more specific towards identification of the structure of your targets, okay, in the human beings rather than the other species. Okay, because there may be pharmacological differences, the interaction of your drug which you are finding out, the lead compound which you have found out, it may change depending upon the species. So you need to optimize your compound based on the activities in the human beings. Okay, so you should be focusing more on the targets in the human beings. Okay, so here to find out those compounds which can very well interact with your target molecules. Now before knowing this, whether your drug can interact, whether your lead compound can interact with your target, blindly simply going for the testing in the animals and human beings, it cannot serve the purpose. It will be simply waste of time. Okay, so there are several assays which have been developed, for checking whether your compounds which you are synthesizing, okay, or you are discovering, so whether they can interact with your target molecule. So there are several in vitro testings which are available. Okay, so here there are some of the examples which are quoted. Cell-free enzyme assays are done, or a membrane-based binding assay is done, or cellular response assays are done. So basically these particular interactions are studied in vitro, and simultaneously a number of tests may be running, number of screening may be done at a time. Okay, so there are simple tests which are available to find out these interactions. Okay, so maybe by utilizing the multi-well plates. Okay, so nowadays a number of studies can be done in using these multi-well plates. So simultaneously robotic assay facilities are also available wherein automated testings are being done. So simultaneously you can test thousands of compound in multi-well plates. Okay, so these are the number of wells which are available in individual plates. So you can do so many studies at a time. So big pharmaceutical industries may be going on for the robotic studies for thousands of compounds which may be run simultaneously to save a lot of time. So you can find out which are those best compounds, best lead compounds which show better interactions. Okay, now apart from this, there are several computerized studies which are also being done. Okay, so these are gaining popularities nowadays, Okay, so your target molecules onto which your drug is supposed to act, so it is being studied in detail, and this target proteins, with the help of computer based molecular modeling, it is very easy to identify, this drug and your target molecule interactions. Okay, so you know what is your three-dimensional structure of your proteins. Supposing if you identify an enzyme which is a target for your drug, so you study that particular structure of that enzyme, so you have the three-dimensional structure of your enzyme available with you. So you can prepare a computer based model for that particular enzyme. So you have the structure of your enzyme. Okay, so computer based interaction of your lead compound with your target molecule can be studied. Okay, so this will in turn help to reduce the work which can be done. Okay, with the help of molecular modeling, okay, you can study the interaction of your drug with your target molecule on a screen, okay, by utilizing the three-dimensional structure of your lead compound and of your target molecules. Okay, now here you can even work out the structures of the derivatives which you have not synthesized them. Okay, so before synthesizing certain compounds, now when you work out a particular scheme, you know which are the derivatives which you can synthesize from that particular scheme. Okay, so what you can do is you can work out the structures of your compounds which you are going to develop. Okay, so hypothetical studies can be done. So these compounds, these are not even synthesized, but with the help of these molecular modeling, you can study the interaction of such kind of molecules with your target proteins. Okay, so you can screen the interaction of your compounds with the target molecule with your target enzyme. Say supposing if you have identified an enzyme on which your drug should act. Okay, so you can work out those different structures of your compounds which you are going to synthesize. With the help of this molecular modeling, you can check the interaction, which are those compounds which can interact with that enzyme in a better way. Okay, so the data which you obtain from here, so you may find, say you may have thousands of compounds which you may synthesize from say particular schemes for chemical synthesis. Okay, so by molecular modeling, you can reduce the number of compounds to be synthesized. So before synthesizing, you can have an idea which of your compounds will be better interacting with your target molecules. Okay, so this will again reduce rather than synthesizing the compound and then checking the interaction in the target molecule. So if you have some means because of which you can check that interaction even before the synthesis, so that will rule out those compounds which are not able to interact, right? So you can find out you can rather select a few number of drugs or few number of lead compounds which show better interaction over here. Okay, so they will, when they show better interaction, obviously they will be predicted to have appropriate pharmacological activity also, right? Okay, so with slight modification in the structure, computer based studies are done to identify which are those lead compounds by interacting with the target molecules with the help of computer based molecular modeling. Okay, so this will again reduce the synthetic work. Okay, now from these computer based molecular modeling, you identify the certain molecules which show best interaction with your target molecule. So such compounds are known as hits. Okay, so by initial screening, you have identified few of those compounds which may have desirable property or which may interact with your target molecule in a better way. Okay, but all of these compounds, lead compounds, may not show ideal property. Some of them may have excessive molecular weight or polarity. Okay, or some may have certain groups which may be responsible for the toxicity. Okay, so several such type of studies have to be done to identify the best lead compound. Okay, so some computational pre-screening of certain compounds is supposed to be done to eliminate such type of compounds, inclusion of the such type of compounds which may not have desirable property as a drug. Okay, so the hits which you have identified from this preliminary screening, computer based molecular modeling, so these are used as a basis to synthesize a number of homologous derivatives, okay, with the help of combinatorial chemistry. Okay, so you critically study the structure of your chemical compound and you can synthesize several such derivatives which will have ideal properties. So you can modify the structure in such a way that all these possibilities may be ruled out, and simultaneously you can develop a number of homologous compounds, okay, which could be future drug. Okay, so this will take you to the next step of the study of your drug discovery. Okay, so such type of repetitive syntheses of certain compounds by slight modifications have to be done, so that best compounds which can interact with your target molecules can be synthesized. Okay, so it's once you have done your computer based molecular modeling, that doesn't mean that particular compound is your final drug which can interact with your target molecule. So you need to basically study whether the ideal compound has been synthesized, or a compound with ideal properties have been synthesized. So a number of syntheses have to be carried out. Again, further screening is required to be done to identify an appropriate candidate for the next step. Okay, now apart from these compounds which are obtained with the chemical synthesis, a particular compound which is obtained from the natural source can also act as a lead compound. Okay, not only the drugs which are synthesized in the laboratory, but there are certain drugs which are also derived from the natural source. Okay, so earlier in the history of the pharmacology, we have seen that in the olden days several natural compounds were utilized as drug, right? So at that time, proper knowledge about the structure or their chemical interaction or interaction with the biological molecules was not known. Okay, or exactly what is that component which is responsible, that was also not known. So a lot of natural products were acting as drug, okay, and they were successfully being utilized as a drug in treatment of certain conditions. Okay, so not only the synthesis of new compounds, but already existing natural products can also act as the lead compound from which you can derive new molecules which may be promising even more than the those which are obtained from the chemical synthesis. Okay, say for example in the olden days the fungal or plant sources were quite utilized, okay, as the therapeutic agents. They were found to be promising in the treatment of several conditions, specifically in the field of anti-infective, okay, that is treatment of certain infections by microorganisms, or in the cancer therapy, or as immunosuppressant drugs. So several naturally obtained substances were also being utilized. Say for example, the successfully utilized drugs which are still being marketed, these include penicillin, streptomycin. There are several other antibiotics also which are derived from the microorganisms itself, vinca alkaloids, paclitaxel, cyclosporin and so on. So these are quite popularly utilized drugs which are obtained from the natural source. Okay, now these fungi, these are found to be quite advantageous. They are quite suitable for the production of the drugs, because these are highly diverse and these are easy to collect and to grow in the laboratory. But there are several disadvantages of the compounds which are maybe obtained from the plants, animals or marine organisms, because commercial production of such type of compounds may not be very easy. Okay. Apart from that, these may be complex molecules. Okay, so their structure may be quite complex, so they may be difficult to synthesize or purify or to modify for the conventional actions, okay, or for a better action. So modification of the structure may not be that easy with the compounds which are obtained from the natural source, and such type of processes, they are purification processes and all, they may be quite costly, so that it will make that drug economically costly. Okay, so it is not ideal. You should go for a drug which is quite cheaper also, right? Okay, so cost is also an important point in selecting a particular drug. Okay, so your processes of synthesis of your drug should not be much costlier, otherwise that will lead to the economical burden on the person who is taking that particular medication, right? Or other people may not prefer for the expensive medication. So that is also an important point which is supposed to be considered. So the problem with these natural sources is their complex structures or their complex constitution. One particular compound may not be present, but a number of compounds may be seen in a particular natural source. So along with your drug, there are several other compounds which you need to isolate, separate. So these techniques may be quite costlier, okay, in the drug development. Then the next step is the lead optimization. So now you have identified the target. Next step is your selection of your lead compounds. Okay, now next step is to optimize the lead compounds. Okay, so here you need to test the potency of your compounds on the target, how better it can interact, how better it can show the action on your target molecules, and to optimize it with respect to certain other characteristics like selectivity. So you should be selectively acting on these targets with a minimum action on the other systems of the body. Its pharmacokinetic properties have to be studied. Okay, so if your drug is having very good activity, but if it does not have ideal pharmacokinetic property, it is quite difficult to administer such type of drugs in the human beings, right? So other properties are also important. No doubt its interaction with your target molecule or showing a particular action on the target molecule is important, but simultaneously its selectivity, its efficacy, their pharmacokinetic properties, these are also equally important. Okay, so you need to optimize your lead compounds. So in this phase, a particular tests are carried out. So certain assays are developed to measure certain activities. Okay, so in these particular studies, maybe in vivo studies also have to be carried out in the animal models. Okay, or to check the unwanted effects of your drug in the animal. Such type of studies also may be carried out, or to check whether your drug is genotoxic, whether it is causing any genetic toxicity. Okay, or the oral bioavailability of the drug may also be studied, because that is also important, right? So if the bioavailability of your drug, it is not good, obviously it will not reach its site of action, right? Now as a target study, it is quite easy. You have your target ready with you and you interact your drug, but actually when you are administering your drug, that may not happen, right? So kinetics are also important to take your drug towards that target molecule, right? So that is equally important. So any drug must have very good kinetic properties, plus it should have less of adverse effects also which it produces. So that is also quite important. So lead optimization is quite important in selecting one of the few lead compounds which you might have identified. Okay, so here again certain lead compounds which you have identified, they may rule out because they may not have appropriate kinetic properties. There are several drugs which have very good interaction, but just because they do not have ideal kinetic properties, they have been ruled out, or they cannot be used for that particular condition. Okay, so these are all the steps in the drug discovery. Now you have a candidate with you which can show effectiveness against a particular condition. Okay, so you know that particular compound can interact with the target molecule for which you have developed it. Okay, now next step is the preclinical studies. Okay, so here you need to test your drug in the animals exactly to study the whole of the pharmacological profile of your drug. Okay, so before you can introduce your drug into the human beings, as I've told you earlier, directly you cannot go for the studies in the human beings. You need to first study your drug in the experimental animals. Work out the entire pharmacological profile of your drug in the animal studies, and if it is found to be favorable, then you can go for the clinical trials. Okay, so here the experiments are done specifically in the rodents, that is mouse, rat, guinea pigs. Okay, and later on the drugs may be studied in the larger animals also, that is cats, dogs, or monkeys or primates. These are quite closer, their physiology is quite closer to the human beings. Okay, so such type of experiments can be done. Okay, preliminary studies are done in the rodents, and then closely related animals are used for the studies of the drug to evaluate the pharmacological profile of that particular drug. Okay, so again here several compounds may again be knocked out in this particular stage also of the preclinical studies. Okay, so such compounds which are found to be interacting with your target molecules, which are found to be very much effective in acting on the target molecule, but such compounds may fail in the preclinical studies also. They may not show effectiveness actually in vivo when they are tested in the animals. So several of the drugs are again ruled out in this particular step also. So very few compounds pass all these preclinical studies and reach the clinical trials. Now let us see what exactly is done in these preclinical studies. Okay, so first of all, screening tests are done. So here simple and rapidly performed tests are done to identify the presence or absence of certain pharmacodynamic activity. Say for example, it's analgesic activity or hypoglycemic activity. So several compounds may be screened for these activities for which it is supposed to be utilized. Say for example, a drug is supposed to be used to reduce the pain, so analgesic activity is carried out over here. Okay, so here whether it shows that property or no, that you have to check first. Okay, so simpler tests are utilized for that. Next, the tests on isolated organs or bacterial cultures may be done. So these are the preliminary tests to detect the specific activity. Say for example, whether your drug has antihistaminic property or anti-secretory, whether it can reduce the secretions, or it has a vasodilatory property or antibacterial property. So this can be studied on by utilizing isolated organs also, which are isolated from the animals. Then the tests on the animal models of human disease can be done. So here you can test whether the drug is effective against a particular situation. So if you have developed a drug, say which can reduce the blood pressure. Okay, so what you can do is you can induce hypertension in the animals and then administer your drug and check whether it can lower the BP. Okay. If your drug is supposed to be active against epilepsy, so what you can do, induce epilepsy in the animals and then administer your drug to those animals. So if it can reduce the seizures, it is effective against epilepsy. Okay, so such kind of tests are done in the animals. So there are several models which are available even for one particular activity. There are several ways by which it can be done, and several species of animals can be utilized for that particular study. Okay, so these are few of the examples which are quoted of the animal models: kindled seizures in the rats, or spontaneously hypertensive rats, or experimental tuberculosis may be induced in the mouse, or alloxan induced diabetes in the rats or dogs. So here certain chemicals may be utilized or certain procedures are utilized to induce those diseases, human diseases in the animals. And such type of animals, these are known as animal models for that particular disease. So after the inducing such situations, you can administer your drug to the animals and check if it is effective. Then next is the confirmatory tests and analogous activities. Okay, now supposing if you know that a compound is effective against a particular pathological condition. Now when you know the pathology of that disease in detail, if you know that certain additional properties, okay, can confirm that particular activity, so those tests are also done. Say for example, when you know your drug has analgesic property, so it is tested for antipyretic, anti-inflammatory activity also. So it confirms the activity upon that particular drug. Okay. Next is systemic pharmacology is studied. Whether your drug is acting on other organ systems, like say how it is acting on nervous system, what are the effects on the cardiovascular system or respiratory system, or on the kidneys or GIT, what are those effects which are shown by the drug on the major organ systems. Okay. Apart from this, the mechanism of action could be studied in the animals, okay, or certain additional mechanisms. Say for example, beta adrenergic blockers, these are basically acting by blocking the beta receptors. So they have antihypertensive property, they can reduce the BP. But additionally, these can also block the alpha receptors also. These may also block the calcium channels also, or vasodilatation may be produced. So some additional mechanisms also may be involved which will add to this activity. So such type of effects are studied in the animals. Some additional mechanisms may be studied. Next is the quantitative test. So now here you can relate the dose with the response which is produced. Okay, how much of dose is required to produce a particular amount of response, or the maximum, what is the maximum effect which can be produced. Okay, or you can compare the potency or efficacy of one drug with another drug, okay, to be sure of whether your new compound is showing activity or no. You can compare it with already existing drug. Okay, the next is pharmacokinetics may be studied. Okay. Absorption, distribution, what are the patterns of distribution, what are the different metabolic processes, how the drug is excreted, what is the volume of distribution, half-life, all these parameters can be studied. Okay. So to find out whether that drug is suitable or whether it is having ideal pharmacokinetic properties also, because this is also equally important. Then another important test which is done is the toxicity testing. Okay, so you need to be sure of the safety of your compound before you go for the human trials. Okay, so here the toxicity testing may be done in more than one species, or at least 2 animal species are utilized over here. Okay. Now toxicity studies can be divided into different types. Okay. Acute toxicity testing is done. So here only a single dose is administered to a group of animal and they are observed for a few days. Okay. Maybe to identify certain appearance of the toxic effects, or maybe this is the best example of this acute toxicity. LD50 is calculated. That is, a particular dose is administered, a single dose is administered to that particular animal, and it is found out at which particular dose 50% of the animals are dying. Okay, so here single dose is administered, and further you can carry out histopathological studies, whether to check for the toxicity of your drug, whether it is affecting the vital organs or not. Then subacute toxicity testing are done, wherein repetitive doses are administered to the animals for a period of say 2 to 12 weeks. Okay, so for example, ED50 calculation or LD50 calculation. So here doses are selected. So again here other actions are also checked. Okay, like what are the food intake, changes in the food intake, or body weight, how it can be altered. Okay, or their growth, how it can be affected, or hematology can be studied, or organ toxicity can be studied. Various organs are isolated and checked for the histopathology, whether any kind of damage is done to those organs. The next type of chronic toxicity is a chronic toxicity. That is, here the drug is given repeatedly for a longer period of time. That is 6 to 12 months maybe this study will be carried out, wherein a repetitive administration is done, and again all the parameters are studied just like which are done in the subacute toxicity. Okay, and when the drug is found to be safer, you can go for the clinical trials. Apart from these toxicity studies, there are specific toxicities also which are studied. Say for example, how it affects the reproduction, or whether it is having teratogenicity, whether it has the tendency to cause the effects in the baby which is growing in the mother's womb. Okay, so how the reproduction and teratogenicity is affected. Okay, whether your drug is having teratogenicity or whether it is affecting the reproduction. Say for example, your drug, whether it is affecting spermatogenesis, that is sperm production, or ovulation, that is release of the ovum, okay, or fertility, or the development of the fetus, okay, whether it is getting affected by your drug. Then mutagenicity can be checked, whether your drug can induce some genetic damage, that is also assessed. Specifically certain in vitro tests are available. AINS test is done for that in bacteria, or mammalian cell cultures are utilized, or sometimes in intact rodents also it is checked, okay, whether there is any genetic alterations which are done. Then again carcinogenicity, that is long-term testing is done to find out whether your drug can induce the tumors in specific animals. Okay, so here basically in the preclinical studies, animal studies are being carried out. Okay, now while you are carrying out animal studies, you need to follow the good laboratory practices. Okay, there are several guidelines which have been laid down. Okay, while carrying out toxicity studies, you need to follow the OECD guidelines. Okay, you should be handling the animals with utmost care. Okay, so there are certain guidelines which needs to be followed while carrying out the toxicity studies. GLP needs to be followed, okay, for utmost care of the animals. Okay, then next step under preclinical trials is the chemical and pharmaceutical development. Okay, now when you have tested your drug in the animals, now you know whether your drug is safe or no. If it is found to be safe, then and only then it will go for the next step. Okay, if it is not found to be safe in the animals, it is rejected from the study. Okay, now supposing those candidates which you have found to be safe, you need to go for the clinical trials. You need to test them in the human beings. Now for these chemicals to be used in the human beings, you need to formulate them, right, into some formulations. Okay, so you need to develop them into certain suitable formulations, right, for them to be ideal to be used in the human beings. Also you need to assess the large scale synthesis also, or purification, whether it is feasible or no. That also you should check. Just finding it to be safe is not enough. Whether you can scale up the preparation of that particular compound, that is also equally important, right? Okay, it should be feasible for you to produce that particular compound in large amounts, or if it is obtained from a natural source, purification should be easy. Okay, also it is necessary to check the stability of your compound, right, whether it is stable over the years. It's not that drugs are prepared and immediately they are consumed. They need to remain stable in that particular formulation for years together, right? Okay, so any company will synthesize a particular formulation, okay, but if it is expiring in 1 or 2 months, it is not affordable, right? Your compound needs to be stable for years together. Okay, so that stability of your compound also is supposed to be assessed. Okay, then the next step in the new drug development is the clinical trials. Okay, so you have discovered a new drug, you have identified suitable leads which can interact with your target molecules. Next, you have tested them in the animals for the safety, for their suitability, whether exactly they are having that efficacy against that condition or not. So when you have done with all these, next step is the clinical trials. Okay, so now here you can test your compounds in the human beings. So once you ascertain that your compound is deserving trials in the human beings, you have to make an application to the regulatory authorities. Okay, so these regulatory authorities will issue you a investigational new drug license for you to test these particular compounds in the human beings. So these clinical trials, these are carried out in a phased manner. It's not that you can directly, when it is found to be safe in the animals, it's not that you can just go on administering this drug to a large population of human beings. Okay, it's not like that. Okay, so to minimize any kind of risk, because you just cannot extrapolate any effect which is shown in the animals to the human beings directly. You need to find out whether exactly it is showing effectiveness. So all these parameters which you have studied in the animals now have to be studied in the human beings, whether actually it is effective in the human beings. Okay, because the species is changing, right? So there may be variations. So there are compounds which may show effectiveness in the animals, but they may not show effectiveness in the human beings. So you need to find out that. Okay? The compound which is found to be safe in the animals, not necessarily it will be safer in the human beings. It may start precipitating some dangerous side effects also. Some life-threatening side effects may also be precipitated. Okay, so to minimize any risk, initially these drugs are tested in few subjects. So it's a phased study which is done. So initially, in earlier phase, you test these compound in only in few individuals, and that too under supervision. You just cannot select a few individuals, go to their house and give the drug to them. No, it cannot be done like that. It should be done in close supervision. So you need to call for these subjects in the clinical trial setup. Okay, usually these subjects are monitored continuously for the types of effects which are shown by your drug. Okay, so later on when it is found to be safer in these subjects, again the number of individuals are increased. So you can go for a larger population. Your drug can be tested on larger number of individuals. Okay, so here again there are certain guidelines which needs to be followed. So what are those appropriate design of your study? Okay, or there are certain ethical points which you need to keep in mind, or how these tests are supposed to, or how these trials are supposed to be conducted. Monitoring is supposed to be done, auditing is supposed to be done, recording or analyzing of the data, and also reporting of these clinical trials have to be done. So what are those ways and means by which all this can be done? All these are put up by the International Conference on Harmonization. Okay, so certain guidelines, okay, have been put forth by the ICH. Okay, so good clinical practices, these are the certain guidelines which will tell you how exactly you should go about the clinical trial. So you just cannot select any procedure or any design and go for it. Okay, so certain guidelines have been laid for carrying out these clinical trials. Okay, so you need to follow these good clinical practices also, okay, to carry out the clinical trials appropriately. Okay, so there are several national agencies in most of the countries. Now, for example, in case of India, it is ICMR, which has laid certain ethical guidelines for the clinical trials. So any investigator who is studying a particular drug in the human beings need to adhere to all these guidelines and need to present or need to record all the data and present it appropriately. Okay, also confidentiality also have to be maintained. Okay, consent of the subjects also has to be taken, okay, as per the guidelines which have been laid down in the Helsinki Declaration of the World Medical Association. Okay. So all such requirements and the regulations for the conduct of clinical trials also has been laid down in the schedule Y of the drugs and cosmetics rule. So need to follow all of these, all the guidelines, and then carry out the clinical trials. Now these clinical trials are basically divided into 4 phases. Phase 1, which is known as human pharmacology and safety. So here human pharmacology and safety is tested. Phase 2, wherein therapeutic exploration and dose ranging is done. Phase 3, wherein therapeutic confirmation or comparison is done. Then phase 4, which is the last phase, that is post-marketing surveillance or post-marketing studies. So these are the phases into which the studies are carried out in a sequential manner. Okay, so let us take up all of these one by one. So first one is a phase 1. So human pharmacology and safety is being tested in this particular phase 1. Okay, so here this is the first human administration of the drug. Okay, once you identify the lead, once you test it in the animals, this is the first phase wherein you expose your drug to the human beings. So this particular administration is done by qualified clinical pharmacologists or trained physicians, and this particular administration is done in a proper setting wherein all the vital functions of those particular individuals can be monitored, and if at all there is any kind of risk or any kind of emergency, okay, all those facilities are available at the hand. Okay, so that if at all there is any deleterious effects, it can be easily controlled or it can be easily treated. Okay, so here the subjects which are selected, these are mostly healthy volunteers. So phase 1 is generally carried out on the healthy individuals. This is rarely carried out on the patients. So mostly there are healthy individuals who are not suffering from any kind of disease. Supposing even if your drug is supposed to treat say diabetes, here diabetic patients are not selected, but first the drug is tested on the normal individuals. Okay, so here usually the number of subjects are also less. Generally it is carried out on 20 to 80 individuals. Normal healthy individuals are involved, and the dose of drug which you select over here, it should be lowest estimated dose, and gradually the dose may be increased stepwise, okay, to achieve or to find out that particular dose which is effective. Okay, so here basically in this phase mainly emphasis is done on the safety of the drug, how safe your drug is, how better the tolerability of that drug is, whether it is precipitating some kind of untoward effect, okay, or to find out what are the kind of effects which is showing on the vital functions. Okay, say for example blood pressure, what is the effect on the blood pressure or heart rate, whether it is increasing or decreasing, whether arrhythmias are getting precipitated or bronchospasm is being induced by your drug, or seizures are precipitated, how it is acting on the kidney or liver, whether it is causing any harm to the kidney or damage. So all your vital functions are continuously monitoring. Okay, so these are all the vital functions are continuously monitored to find out whether your drug is showing any kind of adverse effects over here or untoward effects in the normal individual. Okay, so such kind of effects, if at all they are produced, these are observed and these are recorded. Okay, again apart from these pharmacodynamic effects, pharmacokinetic parameters of the drug in the human beings is also. So here no blinding is done. That means the individual or the volunteer as well as the physician, both of them know that the particular patient is being administered with the drug under the study. Okay, blinding means they are kept unaware. Okay, they do not know whether that formulation which they are consuming, it actually contains that drug under study or no. That is known as blinding. Okay, so here it's a open label study. Okay, so that means here no blinding is done. Okay, both of them are aware that it is a drug which is being administered. And the next phase is phase 2, wherein therapeutic exploration and dose ranging is done. So this is usually conducted by the physicians who are trained as clinical investigators. Okay? And basically these tests are, or these studies are done on a larger number of individuals. Okay? Generally 100 to 500 patients are selected, okay, for the studies to be carried out. Now main aim of this phase is to find out whether your drug is therapeutically efficacious or no, whether it is showing effectiveness against a particular condition or no. Okay, supposing if it is discovered for lowering the blood glucose level, whether it is actually lowering the blood glucose level or no. So therapeutic efficacy of your drug is tested over here, or the dose range in which it shows this effect is also studied over here, or what is the ceiling effect. Okay, what is that maximum dose which shows this particular kind of effect. So that is also checked over here. Okay. So here the tolerability and pharmacokinetic studies which are started in phase 1, these are extended to the phase 2 also. Okay. So generally here this particular kind of study, it may be blinded. Okay. So here randomly selection of your individual subjects is done and blinding may be done. That is, either the physician who is carrying out the studies or the patients may not be aware of whether they are taking the medication or no. Maybe certain individuals may be administered with the actual drug, whereas in certain individuals placebo may be given, which does not contain any active ingredient, or your drug under test may not be present in that. Okay. Or it may be an open label study. Okay. That is, no blinding may also be done. And usually these kind of phase studies are done on more than one centers. Okay. That is, at say 2 to 3 centers in different areas, such kind of studies may be carried out. Okay. Then next, if your drug is found to be effective, so far up to phase 2, if the drug is found to be efficacious, then it should go for the phase 3 study. Okay, otherwise the drug may be ruled out or knocked off from that particular phase itself. So many of the drugs get rejected out because they do not show effectiveness in the human being. Sometimes it happens that if they may show effectiveness in the animals, but they may not be effective in the human beings also. Okay, so likewise many of the agents may be knocked out at phase 2 itself. Okay, some at the phase 1 itself. Okay, so those who are found to be efficacious, those drugs only will move towards the phase 3. That is, therapeutic confirmation and comparison is done over here. So here these type of studies are randomized double blind comparative trials basically. Okay, so here both the physician as well as the patient may not be aware, okay, which of the formulation contains the drug. Okay, so here the studies are also done on the larger population. Okay, large number of patients are selected for these studies. Say around about 500 to 3000 patients may be selected. Okay, by several physicians may be testing these particular drugs in a number of individuals. Okay, at a number of centers. Okay, not only 2 to 3 centers, but many centers may be carrying out such type of studies simultaneously. So here the aim of this particular phase is to establish the value of your drug in relation to existing therapy. So here basically the comparison is being done. Now there are a number of drugs which are already marketed, right, for the treatment of hypertension, for the treatment of diabetes. So if you have developed a new drug which is effective against diabetes, so you need to compare it with the existing therapy, right, how it is better, whether it is better or no. If it is not having any advantage over the existing therapy, then there is no point in bringing that drug in the market, right? Nobody will prefer that. So that is basically established in phase 3. Okay, so here safety and tolerability again, which we have tested in the phase 2. So these are assessed on a wider scale. Okay, whereas the pharmacokinetic studies, these may be continued in few individuals, a few participants of these study, okay, to establish a proper pharmacokinetic data. Okay, so whatever findings done in this particular phase. Okay, these are submitted to the licensing authority. So a new drug application, it is submitted to the licensing authority. Okay, if your drug is found to be safe, and if there is therapeutic confirmation, and if it is found to be better than the existing therapy, you can file a new drug application to the licensing authority. So if they are convinced with your data, okay, that if it is better, then they can give you permission for marketing of that particular drug. Okay, so then the drug is released in the market in the controlled manner. Okay, so that is known as the phase 4, post marketing surveillance or study. Okay, so phase 3 is over. That doesn't mean, if your drug is found to be safe in phase 3, that doesn't mean that it is completely safe and it can be released in the general population. So it's not like that. So after that, when the drug is released in the market, so over there again general testing of your drug is done over a wider range of population. Okay, so here the drug is released in the market for general use. Okay, certain practicing physicians are identified and data is collected from such physicians. They will prescribe the drug, okay, which is found to be safe in phase 3. They will prescribe such drugs and they will collect the data from the patient about the efficacy or acceptability of the drug or adverse effects. Certain drugs may have long-term adverse effects which may not be detected during the clinical trials. So such data is collected from these selected physicians and it is recorded. Okay, so here since you are releasing these particular drugs on a wider scale, so here the number of population which is studied is still larger, okay, as compared to the earlier stages which we have seen, phase 1, phase 2 and phase 3. So over there a limited number of population were being administered with the drug, but here a much larger number of population is involved in this phase 4. Okay, so here your main aim should be to study the long-term effects of your drug, uncommon adverse effect or idiosyncratic adverse effect which are characteristics to certain individuals. So these can be studied in the phase 4. Okay, so here you may get an appropriate idea about the drug when it is prescribed in a wider range of population. Okay, so whether there is some kind of variation in the effects which may be seen, because so far you have selected certain individuals, right, and in those you have tested your drug, but here the physician is prescribing the drug to the general population. So here it is better understood the adverse effects, the long-term adverse effects in general use, it is identified. Whether there is any kind of drug-drug interactions which are possible, that can also be studied in the phase 4. Okay. Then after this, all these studies being carried out, there are certain special cases wherein the drug might have to be studied. Say for example, in case of children, the studies have to be carried out. Elderly or pregnant or lactating women or patients with renal or hepatic disease. Now all such kind of individuals are not involved in the clinical trials phases which we have seen so far. Okay, so such type of specialized studies also may be undertaken to find whether the drugs are safer in children, because children are not small adults, right? In them the physiological processes are altogether different. Even in case of elderly, even in case of pregnancy, okay, or lactating women, okay, it may affect the fetus or the suckling infant. So you need to study over there. Again, if the patient is suffering from renal or hepatic disease, the disposition of the drug will get affected. So you need to study your drug pharmacology in such individuals also, whether what kind of or how your drug action is getting altered in all these situations. Okay. Again, the modified release dosage forms. Okay, or some additional route of administration. Maybe in the clinical trials you have given your drug in the simple dosage form, say for example in the form of a tablet which is supposed to be administered by oral route. Okay, but supposing if you want to release that same drug in the form of an injection. So again, additional studies have to be done, right, when the drug is given in the form of additional type of dosage forms, or some other routes of administration are used, or supposing if you have to combine more than one drugs together, so fixed dose drug combinations, so such type of drugs may have to be also explored additionally. You just cannot apply the study clinical trials of individual two drugs to the combination of the two drugs. They may act differently, right? Okay, so those studies, those type of studies also have to be done additionally. Okay, even supposing if your drug is found to be safe in phase 4 also, okay, and if it is allowed to be prescribed in general population, okay, on say larger scale, even more larger scale, still the drug is continuously being monitored. There are several companies which are involved in the vigilance, okay, which are continuously monitoring the adverse effects of the drugs. Okay, so it's quite important because there are certain examples of drugs which can precipitate adverse effects even after years of administration. Okay, that is after several years of marketing, there may be certain adverse effects which may be identified. There are examples of drugs which have been withdrawn from the market after years of prescribing. Okay, so that is also possible. So continuous monitoring of your drug, so you can say that your drug, even though it is marketed, it is under a continuous development process, okay, because you never know, a particular drug may have a long term adverse effects also. Okay, so continuous monitoring of your drug actions and effects is very, very important. Okay, so here in this picture we can summarize the process of drug discovery, preclinical development, and the clinical development. So how a new drug is developed exactly, so all the processes are summarized what we have seen. So first step is the drug discovery, wherein you select a target, find a lead compound, lead optimization is done, and then pharmacological profiling of your drug is done. So usually hundreds of such drugs or such chemicals may be studied, okay, for the new drug development, and this process may take around about 2 to 5 years. Okay, so here after the drug discovery you have identified a drug candidate. Okay, so such kind of compounds, these are first studied in the animals, okay, or preclinical development you can call it. Okay, so here pharmacokinetic or short term toxicity, formulation development is done, or whether synthesis scale up is possible. Such kind of studies are done in the preclinical development. So this process usually takes 1.5 years time. Okay, so round about maybe say 100 compounds were discovered, okay, in this particular stage. Out of that maybe only 20 compounds were found to be suitable, which can go for clinical development. So here onwards the development of your actual drug starts. So here the drugs are being tried in the human beings. So here kinetics, tolerability, side effects in the healthy individuals are tested. So say for example, out of the 20 compounds which are found to be ideal in preclinical development, out of that only 10 compounds will be found to have ideal properties in phase 1, which will enter the phase 2. Okay, say for example, out of this only 5 out of the 10 compounds only 5 have reached the phase 2, or they have successfully completed the phase 2, that is small scale trials in the patient, okay, to check the efficacy and dosage selection of your particular drug, or sometimes long term toxicity testing is also done in phase 2. Say out of these 5 only 2 may reach the final outcome in the phase 3. Okay, so that is large scale controlled clinical trials which are carried out, and the process of all these phase 1, phase 2 and phase 3, it may take around about 5 to 7 years for the compound to reach its final phase 3. Okay, following which, if it is found to be safer, all the data is submitted to the regulatory authorities for approval. Okay, so once these regulatory agencies they approve your project, that particular process may also take around about 1 to 2 years. Okay, it's not that when you submit the data immediately you will get the permission. No, it may not happen. Okay, so that process also takes quite a long, and maybe out of 2 only 1 may get a permission for the phase 4 trials, that is post marketing surveillance. So here you get approval for the marketing of your drug, that is you can actually make your drug available on retail base to the general population. Okay, so that is known as the phase 4. Okay, so this may be continued for years together to study the long term adverse effects of your drug. So you can say that the development of a new drug, it's not a 1 or 2 years job. Okay, a particular drug may take around about 15 to 20 years for it to come into market. You can see the number of years which are involved in each step. Okay, so a particular drug may take several years to come into market, and that too after coming into market also certain drugs may get rejected. Here you can see that we had started with hundreds of compounds, but out of those 100 compounds only one has reached the market. So at several steps the drugs may get, or the compounds may get rejected. They may not be ideal. Okay, so you will find it out in the later stages of your trials that the drug are not ideal, okay, which were found to be promising in the drug discovery. It's not necessary that same drug will reach phase 4. Okay, so this is to do with the drug discovery and the clinical trials, okay, or the clinical development of a drug. Now the important question relating to this topic is note may be asked on the drug discovery, or a note on clinical trials may also be asked. Thank you.